The previous lecture discussed anaerobic metabolism, so this one will cover aerobic metabolism. So this lecture will break aerobic metabolism into two parts, Krebs cycle and the electron transport chain, and the topics and outcomes are outlined here. To start off, the Krebs cycle occurs in the mitochondrial matrix, which is essentially the fluid inside of the mitochondria. The Krebs cycle is an 8-step process that begins with a molecule called acetyl-CoA. While oxygen is not required for the Krebs cycle, it is important that oxygen is available. If oxygen is available, then pyruvate will be created at the end of glycolysis, which can then be converted into acetyl-CoA. If oxygen isn't available, then lactate will be formed, which means acetyl-CoA will not be formed. Now, each molecule of acetyl-CoA can go through one full cycle of the Krebs cycle, which results in the formation of one molecule of ATP, as well as some carbon dioxide, NADH, and FADH2. The carbon dioxide needs to be eliminated, which we do by breathing out, while the NADH and FADH2 will be used later in the electron transport chain. Since we get two molecules of pyruvate from glycolysis, this allows the formation of two acetyl-CoA. Each of these go through a turn of the cycle, and ultimately we form 2 ATP. Isocitrate dehydrogenase is the rate-limiting enzyme for the Krebs cycle. This is the enzyme that's responsible for the third step, just like phosphofructokinase, interestingly enough. And just like phosphofructokinase, isocitrate dehydrogenase is sensitive to ATP concentrations, such that as ATP concentrations are high, which means we have a lot of available ATP, isocitrate dehydrogenase activity decreases. And the reverse is true. In other words, we've got another example of a negative feedback loop. So at the end of glycolysis and Krebs, we go on to the electron transport chain. Now, the electron transport chain occurs as molecules cross the inner mitochondrial membrane. Okay, the membrane of the mitochondria has an inner and an outer membrane. And so if we look here, oh, that's not a very good color. This is what I was looking for. So here is our outer membrane, and here is our inner membrane. And this is what we call the inner membrane space. All right, so the electron transport chain occurs as molecules cross the inner mitochondrial membrane using a series of proteins and chemical reactions. Okay, so simply put, NADH and FADH2, which were created during glycolysis and Krebs, carry electrons and hydrogen ions to the proteins involved in the electron transport chain. And if you look, we've got a series of 4 proteins here. Okay, so what happens is electrons and hydrogen ions move, all right? And this causes NADH and FADH2 to be converted to NAD and FADH, which then can be reused in other steps of cellular respiration. So what happens is these electrons from NADH and FADH2 are passed down the chain. They move from a high level of energy to a lower level of energy. And in other words, they release energy, okay? And some of this energy is used to pump hydrogen ions. So think of these proteins as pumps, right? And so we've got a pump here. So we're pumping hydrogen into this space. Same with here, same with here, okay? And so what this does is this actually establishes a gradient. At the end of the electron transport chain, electrons are transferred to oxygen, okay? And then this oxygen splits in half and combines with hydrogen ions to form water. Well, these hydrogen ions are actually coming through this protein, through this pump called ATP synthase, and they travel down its gradient, okay? And then 2 of them combine with oxygen to form water, okay? Now, as the hydrogen ions flow down their gradient and back into the mitochondrial matrix, they pass through this ATP synthase, which harnesses the flow of hydrogen ions to synthesize or create ATP. Okay, basically what this protein is it's a big axle, and this axle spins around and it smashes ADP and phosphate together. And now we have ATP. Okay. Now, in a perfect world, one molecule of NADH would produce 3 molecules of ATP, while one molecule of FADH2 would produce 2 molecules of ATP. Okay. So ultimately, we have 10 NADH and 2 FADH2 that came from glycolysis and Krebs that can be used to produce ATP. And ultimately, between 28 and 34 ATP are produced, depending on what's going on and the amount of other things that are available. Okay, and the reason I mention this range is just to give you an idea of or just to let you know that there is a range, and to the fact that different textbooks give different values. So you'll see anywhere from 2.5 to 3 ATP for NADH, or 1.5 to 2 ATP for FADH2. Now, there isn't a rate-limiting enzyme for the electron transport chain. However, it is similar in that high levels of ATP inhibit the transport chain. So again, we've got another example of a negative feedback loop. And the electron transport chain is used for activities that last longer than 3 minutes in duration. Okay, because this is the one where we're producing lots of ATP. Glycolysis produced 2, maybe 3 ATP. Krebs cycle produced 2 ATP. The electron transport chain produces about 30. So to help you review, see if you can answer these 2 questions. Explain how the Krebs cycle works, and explain how electrons and hydrogen ions are used in the electron transport chain.